Okay, welcome back. I hope you have enjoyed the lunch. And it is now time, and my pleasure, to present our next speaker. Is Professor Dr. Lunes. Professor Brown, please welcome.
Thank you very much. Nice introduction. So I will take advantage to thank everyone who is attending this meeting. It's really nice to see all students here eager to learn about biomedical sciences. Of course, it is for us a pleasure to have all these great thinkers, great scientists who are coming from abroad and from a real problem, and it comes with some health problems too. I mean, probably you know that the progressive increase in incidence of chronic diseases is probably, at least in part, explained by the global aging process. More importantly, global aging and it has an impact in cancer. Mainly, it's the main risk factor for many of the most frequent cancers in humans. So particularly those that originate in epithelial tissues. How to define aging from a biological perspective? Here's one definition, simple, probably there are other definitions of aging. Aging is the progressive loss of physiological integrity as a result of accumulated cellular damage. What are we understand by the term cellular damage? Here is a compelling hallmarks, a collection of hallmarks of aging that was published in 2013, and probably, as you saw in the previous presentations, there are other accounts, more recent accounts of these hallmarks of aging. I'm not going to go through all of them, just let you know that one of the main hallmarks of aging right now is the process of cellular senescence. Yeah. So how do you define cellular senescence? Simply speaking, is of permanent cell cycle arrest triggered in response to a wide range of physiological and pathological stimuli. So this is the history of aging, of senescence, cellular senescence. As it was already mentioned in the morning, there's a concept called replicative senescence. That was the first phenomenon that was described more than 60 years ago by Hayflick and Moorhead. And basically, this is the basics or the basic concept that senescence is basically due to continuous replication in primary cells, probably triggered by the shortening of telomeres, and ultimately triggering the DNA damage response. Almost at the same time that replicative senescence was described back in the 60s, it was evident that this could be a barrier for tumor progression. So the concept is that to become fully transformed, the precancerous cell must overcome cellular senescence. Yeah. Of course, we know now that senescence can be triggered by several stimuli, not only the shortening of telomeres, and this is called stress-induced senescence or premature senescence. And key among these triggers probably are drugs that are used in the clinic in cancer patients or subject to chemotherapy. And probably you know that many of these drugs already induce senescence as part of their mechanism of action. So I'm not going to talk a lot about the features of the morphological features or the functional features of senescent cells, but there is one aspect that is key in our research, and it has to do with this ability of senescent cells to secrete and release soluble factors that ultimately can remodel the microenvironment. So we know that this feature, which is called the senescence-associated secretory phenotype, we know that this feature can impact at the organismal level at several points. So let me give you some examples. For example, the physiological aging is characterized in many cases by the accumulation of senescent cells and in many tissues. These senescent cells, due to this ability to secrete a lot of factors, many of them pro-inflammatory, can in turn lead to the formation of a microenvironment which is pro-tumorigenic and ultimately lead to the arising of cancer. Another scenario can be, for example, in the case of cancer patients who are subject to chemotherapy and with drugs that induce senescence as part of their mechanism of action. So in this case, one can imagine that these drugs will create senescent cells, and if these senescent cells are not removed, the secretory factors they release can in turn continue to act locally and produce a pro-tumorigenic environment. So this is one slide just to summarize something that Whitney Venturini will talk in the next talk. We tried to, at some point, understand the interaction between senescent cells and platelets, more specifically endothelial senescent cells and platelets. We believed that this interaction is important to explain, at least in part, some mechanism that can lead to thrombotic diseases, which is the basis of most cardiovascular diseases. The idea was quite simple. Basically, endothelial senescent cells are able to secrete factors that in turn can activate and aggregate platelets. And this will be a mechanism that will explain why, for example, older people are more prone to develop thrombotic diseases. As I told you before, you will hear more about this in the next talk. So, one concept, a relatively new concept about senescence is the concept that, at least in tissues, obviously, once you accumulate senescent cells, they can be detrimental for the health of the organism. As I told you before, senescent cells accumulate, their SASP remodel microenvironment and then lead to the development of tumors or other type of diseases. Now, in this context, it is now recognized that the immune system plays an important role in removing these senescent cells in such a way that, for example, in the context of chemotherapies, so you remove senescent cells induced by chemotherapy, and the outcome of reducing the chances of recurrence of the tumor are much better. So in the lab, we are working on this simple idea. Basically, as you see here, when you have a tumor, and this tumor is treated with chemotherapy based on pro-senescent chemicals or drugs, you see an accumulation of senescent cells in situ, which is good for the patients because the tumors won't grow. But if these senescent cells are not removed by some mechanism, the senescent cells there will produce a pro-tumorigenic environment through the ability of senescent cells to secrete chemokines, cytokines, and other pro-inflammatory factors. So the concept here is that for the therapy to be successful, you need to remove senescent cells at some point. You need, and this in a lot of patients is done by activation of the immune system. So we try to address this possibility, the possibility that senescent cells are capable, through the release of these same SASP factors, are capable of activating some components of the immune system. So this is our favorite model here. We work with gastric adenocarcinoma cell line, AGS. AGS is a well-known cell line that is still contain wild-type retinoblastoma protein, and therefore it is still sensitive to some drugs. One of them, very important, which is used in our lab to induce senescence, is palbociclib. Palbociclib is a CDK4/6 inhibitor, and therefore is in theory capable of inducing senescence in cells that are RB positive. In other words, cells that still retain the function of retinoblastoma protein. So one student in our lab, Raúl Segovia, did a simple experiment, tried to check the expression of several factors that were published and are known to be released by senescent cells. He basically did a qPCR experiment checking several factors. Some of them are up, some of them are down here. So we try to conclude from this that many of the factors that are up are really pro-inflammatory factors. Indeed, we were able to show, as you see here, that some of these factors, pro-inflammatory factors, are indeed detectable at the protein level in the conditioned media extracted from these senescent cells. So this slide basically shows that as a model, the induction of senescence in AGS cells really recapitulate what we were expecting, basically a phenotype that is pro-inflammatory in the sense that many of the factors released by these senescent cells, AGS senescent cells, were really pro-inflammatory. So Raúl went one step further. What he did was to characterize these senescent cells for the expression in their surface of some markers that are recognizable by immune system cells, basically NK cells or CD8 positive T lymphocytes or cytotoxic lymphocytes. We did this because we wanted to corroborate that senescent cells or this model of senescent cells really was recognizable by the immune system. So as you see here, he did again a qPCR experiment trying to confirm the expression of several factors that are expressed supposedly on the surface of senescent cells and are recognizable by natural killer cells, which are a type of innate immune system cell. What you can see here below in the lower panel is basically an experiment, one of the last experiments in which Raúl what he did was basically use cytometry to study some of these factors, basically ULP 5, 6, and 2. There was an antibody, lucky us, there was an antibody that recognizes all three forms, and so it allowed us to study the expression by flow cytometry on the surface of senescent cells. And as you can see here, really senescent cells are expressing more of these factors on the surface, and in theory they are then recognizable by NK cells. He also did an experiment showing that there is a small increase in the expression of MHC1 molecules, major histocompatibility complex 1 on the surface of senescent cells. Remember, these molecules are important for the expression of some antigens that are recognizable, are recognized by in this case, T cells. particularly CD8 positive T cells. And he observed that, maybe not surprising, even AGS cells that were not senescence, they were treated only with the vehicle, with DMCO, where also they had plenty of these molecules on their surface. This was not surprised, because all our nuclear cells, including cancer cells, they express these molecules on their surface. So, but he did observe a change of 4 to 5% he observed an increase in these molecules in senescent cells. And also, he observed that the brightness of the staining and flow cytometry was also increased in AGS cells treated with palbociclib. In other words, in cells that were senescent. So the next step was also an experiment that was quite simple, was seemingly simple, but in reality was not simple. In this case, it was to have the supernatant of senescent cells, I mean the conditioned media of senescent cells and non-senescent cells, and use this media to stimulate the activation of immune cells. Now, the immune cells were provided by healthy donors, in which we obtain the human peripheral blood mononuclear cells, the PBMCs. These cells, or this fraction of cells, contains mainly monocytes and lymphocytes. And these cells, once they were isolated, purified, were then incubated with conditioned media provided by AGS senescent and non-senescent cells. And then we subjected the whole population of PBMCs to flow cytometry. What he observed was something that we still are trying to solve and trying to understand. Basically, if you see the quadrant 1, here are mainly the NK cells, and he saw not much difference in quadrant 1 from the analysis using conditioned media from non-senescent cells versus conditioned media from senescent cells. But in quadrant 4, he observed almost a double increase in this cell population. We still don't understand what happened here. In theory, in quadrant 4, you have B lymphocytes. In quadrant 3, we saw a reduction in the amount of cells in the population treated with conditioned media derived from senescent cells versus non-senescent cells. So we still don't understand this. But if we focus on quadrant 1, which is at this point our main interest, because it contains NK cells, Raul observed something very interesting. He observed an increase in this population or subpopulation of NK cells, which are active NK cells. Although the total number of cells is very low, we still saw a double in the increase suggesting that the conditioned medium obtained from senescent cell somehow is capable of increasing the expression of some markers that are sign of activation of NK cells. Interesting, we observe the same for T cells, almost a double in this population. We still don't know what this population of the exact identity of this population is. Probably these are also cytotoxic T cells. Remember that cytotoxic T cells and NK cells share some mechanism of how to kill other cells. They use effector mechanisms that are very similar. So this very preliminary data basically shows that at least to some extent factors released by senescent cells, in this case AGS senescent cells, are capable of changing the proportion of mononuclear cells isolated from healthy donors, and at the same time, at least some proportion of these cells are activated when exposed to this conditioned medium. Now I would like to switch to another cell of the immune system. As I told you before, our paradigm thinking is that for a successful chemotherapy based on pro-senescence drugs, we need to have these senescent cells removed. So another cell of the immune system that has been shown to be able to remove senescent cells is monocyte macrophages. Remember, monocytes are circulating cells that are derived from the bone marrow, and then these cells, when they enter the tissues, they quickly differentiate into macrophages. So, is it possible that these macrophages can stimulate, or better say, they can act to tumor cells? So there is a student, undergrad student in our lab, Matias Mesa, try to understand almost by chance, try to understand what happens with monocytes. In this case, monocytes that are subject to differentiation and at the same time exposed to palbociclib. First, before showing the actual results, let me tell you that the differentiation of macrophages is more complex than we thought. Remember, monocytes can differentiate into basically two types of macrophages, M1 macrophages and M2 macrophages. These two subpopulations of macrophages have different functions during inflammation, for example. M1 macrophages are considered more pro-inflammatory, whereas M2 macrophages are considered more anti-inflammatory, and they are involved mainly in the process of tissue repair. Now, these two type of macrophages derive from, I would say, macrophages that are called M0 macrophages. And in order for switch to to any of these subpopulations, they have to interact with several molecules, inflammatory, pro-inflammatory molecules, or anti-inflammatory molecules, and then to get any of these populations. So it is quite, in real life, it is a quite complex process. So Matthias asked the following question: how is monocyte macrophage differentiation affected in cancer patients treated with palbociclib? That we know that is a pro-senescent drug. And he used a very simple type of model, again, an in vitro model. Basically, he took these THP1 cells, which are cells that derive from monocytic leukemias that retain the ability to differentiate into macrophages, and treated these cells with palbociclib for 96 hours, and these cells acquire some characteristic of senescent cells. And then he continued to differentiate these cells using PMI and LPS for 24 hours and to differentiate these macrophages into M1 macrophages. Interesting. So here's the result of beta-galactosidase staining. You see that we were able to detect beta-gal activity, and this beta-gal activity was decreasing over time. We confirmed that these cells, even those that were not treated with palbociclib, were not proliferating as they progressed through the M1, M0, and M1 differentiation steps. And this makes sense because these cells, when they are even when they are not senescent cells, the treatment we use, they basically make them exit the cell cycle and enter a differentiation, a terminal differentiation step. Now, there are some very, at the beginning, very striking and difficult to explain results. One of them has to do with the expression of P21. Remember, P21 is by many used as a marker of orchestration of senescent process. So in this case, what we observe, if you take you only see the lanes 1, 3, and 5. So you see all non-senescent cells that as they progress to differentiation, they increase the level of P21, indicating that P21 is also important for terminal differentiation of THP1 cells into M1 macrophages. What was not expected was that senescent cells, THP1 cells, monocyte cells treated with palbociclib showed a reduction in the expression of P21, and this was catch up only later to become fully expressed much later in the fully differentiated senescent M1 cells. So this means that at least in these cells, the treatment with palbociclib interferes with the process of differentiation evaluated in this case by the expression of P21. On the other hand, what we can see with the phosphor PRB, which is the phosphorylated form of retinoblastoma protein, is what we are expecting. So we see reduction in the expression of this isoform of PRB in senescent cells compared with non-senescent cells. So these experiments we are trying to explain and get our heads on this type of results. Maybe it's an indication that what we are seeing here is interference with the differentiation program that also requires cell cycle exit and activation of the RB pathway. Finally, Matthias also checked the expression at the mRNA level of several factors that are supposed to be expressed in M1 macrophages. And what he observed basically was that M1 macrophages, of course, they show expression of these markers when they were not non-senescent, but senescent. M1 macrophages were basically not expressing these markers, which is an indication that senescent M1 macrophages really are not functional, or at least based on these markers are not working as truly differentiated M1 macrophages. Interesting, when Matthias checked the expression of M2 macrophage markers on these cells, he observed some of these markers are retained. Not all of them, but some of them are retained in this senescent M1 macrophages, which is an indication maybe that senescence M1 macrophages by default are expressing M2 markers. And finally, M1 macrophages are characterized by the ability to phagocytosis, to engulf particulate bacteria or other particulate substances. So Matthias, together with Claudio Valenzuela, who is a professor in our lab and who is in charge in the practice of our students, they basically implemented an assay to check phagocytosis of these M1 senescent cells. And I'm not going to describe in detail this process, but basically involves infection with E. coli, which made me very nervous the first time they implemented, because of the risk of contamination, of course. So they basically infected cells, M1 macrophages, either senescent or non-senescent, with bacteria. After 1 hour, they basically started a treatment with antibiotics, and then the cells were subjected to cell lysis, and the viable bacteria were cultured, and that's the way in which you know that the phagocytosis process occurred correctly. So basically what they observed is that in cells and one macrophages which were senescent, they observed a reduction in the ability to finally form polymy units after agar seeding, which means that the process of phagocytosis, not the whole process of killing the bacteria, but only the process of phagocytosis is not working as well in these M1 senescent and M1 macrophages, which is also is a sign that basically senescent M1 macrophages are not functionally acting as normal M1 macrophages. And so this is what we had for you today. So I would like to thank all members of the lab. We are not a single lab, we are several labs together to helping each other. Some members of the lab that are now principal investigators by their own merits. For example, Whitney Venturini, Angel Cayo were no longer students, of course. And I would like to thank to our students, Raúl Segovia, Matías Mesa, Francisca Torres, who is doing a different sort of project, and all our collaborators, and especially to our faculty members, Dr. Rodrigo Mori and Antonio Valenzuela, who are our partners. We are partners with, in this effort. Thank you very much. I don't know if there are some questions.
Thank you, Professor Brown. There is time for some questions.
Yes.
Thanks, Nelson, for your wonderful talk. I have a question about you are using Cellular. Since you are already working with Auditorio, are you planning to use H. pylori, for example, in thinking in gastric cancer, APS,
Yes.
as an alternative or publicity?
Well, that's a good question. I think that would be very nice to do those experiments with Helicobacter pylori. And I'm not sure how easy it is to culture those cells, so those bacteria. Yes, I think that's something we should talk about and try. Now, I forgot to say that the reason we are using a cell line which is derived from carcinoma gastric cancer cells, basically, is because we are also interested in applying all these concepts to the pathogenesis of gastric cancer, which is very is a real problem, especially here in this region. So yes, there are a lot of I would say avenues we can take with these preliminary experiments. One of them is certainly study what is going on, what is the relation of these immune cells with gastric tumorigenesis, and where the senescence part is playing a role there. I mean, we are not entirely sure. I mean, I have my hypothesis, but it's still something we have to work on.
Another question?
Yes.
So if I saw that correctly, you treat with palbociclib the THP cells,
Yes.
and then after that you try to differentiate it and get this result. What do you think would happen if you do it the other way around? Like you have already polarized M1 macrophages and you treat it with palbociclib, like imagine those macrophages are already in the tumor and now the treatment comes, how would it,
Yeah.
have you done it? And if not, what do you think would happen? Any idea of how it would
Yes.
react?
Actually, we have done it. We did, and we, to be honest, there were some results that were very similar to what I showed. In terms of senescence, but we didn't continue in that avenue. We tried to first characterize this type because there were many different combinations. So we would try first to characterize this other model, which begins with induction of senescence. Now, having said that, remember what we are seeing here is, in the case of treatment with Th1 cells, is cells that are still proliferating, and you treat them with palbociclib, they enter senescence. I'm not sure what are we looking at when you already differentiate cells into M1 or M2, for example. The cells already exit the cell cycle, and then you are using palbociclib to induce, I'm not sure, because it's not replicative senescence anymore. I mean, it is something else. So in that case, what probably you are studying basically is the effect of CDK4/6 inhibition in the differentiation process, probably, and then yes, I think those are experiments we have to pursue. Yeah, but we haven't done it, or we don't have so far done it. So I think Matthias has to take note.
So, thank you, Professor Brown, for your excellent presentation. And it's now time to move to the next speaker. Okay, our next session is dedicated to young and talented scientists. Our next speaker is Professor Whitney Venturini from the Department of Translational Medicine, Faculty of Medicine of the University Catholic of the Maule, Talca, Chile. Professor Venturini has obtained her PhD in Biomedical Sciences from the University of Talca under the supervision of Professor Rodrigo. So please welcome Professor Venturini.
hoy les voy a contar un poco un trabajo que llevamos bastantes años realizando, y es cómo podemos nosotros relacionar a la senescencia endotelial con el proceso de trombosis. El cáncer es una enfermedad crónica no transmisible que se caracteriza por defectos en la supervivencia, proliferación y diferenciación de células somáticas. Esto trae como resultados que las células crezcan sin control, es decir, que formen tumores, y en algunas circunstancias estas células pueden invadir tejidos vecinos y formar metástasis. La Organización Mundial de la Salud determinó que para 2021 el cáncer constituía la segunda causa de muerte a nivel mundial. Para el año de 2025, el cáncer pues seguía siendo la segunda causa de muerte a nivel mundial, y las proyecciones que nosotros tenemos para 2026 es que existan más de 2 millones de nuevos casos y que al menos 600 personas mueran producto de esta enfermedad, lo que quiere decir que al menos una de cada 6 personas diagnosticadas con cáncer puede morir de esta enfermedad. Gracias a los grandes avances que hemos tenido en el diagnóstico y tratamiento, hoy nosotros podemos decir que el 80% de los pacientes puede sobrevivir a esta enfermedad. Si nosotros vemos las proyecciones de 2022 a 2040, podemos decir que van a existir más de 8 millones de sobrevivientes de cáncer. Sin embargo, estos pacientes presentan complicaciones a largo plazo y están principalmente relacionados a la terapia que reciben. Por lo tanto, el 37% muere producto de enfermedades cardiovasculares, las cuales pueden estar relacionadas, al menos por una parte, en la disfunción endotelial que pueden producir estas drogas en el endotelio vascular. El cáncer, debido a su alta incidencia, es un problema de salud pública, lo que ha traído como resultado que existan múltiples opciones de tratamiento, las cuales van a tener, por una parte, van a permitir detener el crecimiento y propagación de células malignas, pero por otra parte también van a proporcionar supervivencia al paciente que padece esta enfermedad. Dentro de los tratamientos y las opciones para tratar el cáncer, de interés para la presente charla es la quimioterapia y específicamente la doxorubicina. La doxorubicina es un antibiótico de tipo antraciclina, el cual ejerce su efecto antitumoral gracias a la combinación de múltiples mecanismos. Por ejemplo, la doxorubicina inhibe a la topoisomerasa 2, puede causar directamente daño al ADN y puede también aumentar las especies reactivas de oxígeno, causando muerte celular. Actualmente, este quimioterapéutico es ampliamente utilizado en múltiples tipos de cáncer, sin embargo, tiene una limitación importante que está asociada a su cardiotoxicidad, donde aproximadamente el 10% de los pacientes que reciben esta terapia pueden desarrollar dicha cardiotoxicidad, lo cual, como es de esperarse, limita su uso clínico. Estos estudios de cardiotoxicidad han sido ampliamente estudiados en cardiomiocitos, sin embargo, se sabe muy poco de qué puede pasar con esta cardiotoxicidad y su relación con células endoteliales. Nosotros acá observamos un endotelio vascular, el cual está constituido por una monocapa de células endoteliales. Estas cumplen múltiples funciones en el organismo, como por ejemplo, pueden regular el tono vascular, tienen propiedades pro y antiadhesivas, y una sumamente importante es que gracias a ellas se regula el proceso de hemostasia. Si nosotros observamos cuál es la naturaleza que tiene el endotelio, podemos ver que en condiciones normales, es decir, cuando no hay ruptura, se favorece un estado antitrombótico que va a estar acompañado de vasodilatación, inhibición de la agregación plaquetaria, inhibición de la coagulación, y se va a favorecer el proceso de fibrinólisis. Sin embargo, cuando hay una ruptura del endotelio, esta naturaleza cambia a una naturaleza protrombótica que va a estar acompañada de vasoconstricción, se va a iniciar el proceso de hemostasia primaria, el proceso de hemostasia secundaria y se va a inhibir el proceso de fibrinólisis. Todos estos son sitios específicos, lo que quiere decir que células endoteliales adyacentes van a tener una naturaleza antitrombótica. Es conocido que en un individuo sano las sustancias antes mencionadas se encuentran en perfecto equilibrio. Sin embargo, en un individuo que recibe quimioterapia, estas sustancias se encuentran desreguladas y van a favorecer a que exista una disfunción endotelial, lo cual es un factor de riesgo. Actualmente nosotros sabemos que una interacción inadecuada entre el endotelio vascular y las plaquetas va a favorecer la formación de trombos y por lo tanto la trombosis. Y las plaquetas son estas células que nosotros observamos acá, son células que cuando se encuentran en reposo tienen un tamaño discoide, son bastante pequeñas, tienen un tamaño de 2 a 4 micrómetros, pero a pesar de ser muy pequeñas se encuentran muy abundantes en el torrente sanguíneo, aproximadamente en un individuo sano 150.000 a 400.000 plaquetas por microlitro. Las plaquetas cumplen un rol importante a través de su vía canónica y su vía no canónica. En su vía canónica, cuando el endotelio vascular sufre una ruptura, las plaquetas se adhieren, cambian su forma gracias a proteínas presentes en su citoesqueleto, posteriormente liberan el contenido de sus gránulos, por ejemplo ADP y tombroxano A2, y estos agonistas plaquetarios van a reclutar a más plaquetas y se va a formar el tapón hemostático primario. Adicionalmente, también se han vinculado en una vía no canónica en procesos de inflamación, por ejemplo, a través de la secreción de mediadores proinflamatorios en cáncer, favoreciendo la adhesión de células tumorales al endotelio, y en procesos de aterosclerosis y trombosis a través de una hiperactivación plaquetaria. Hasta ahora se conoce que la quimioterapia puede reducir los factores anticoagulantes, puede favorecer la activación plaquetaria y puede causar daño en el endotelio vascular, lo cual finalmente va a favorecer el desarrollo de trombosis. Sin embargo, el mecanismo de cómo esto ocurre ha sido poco estudiado y se puede explicar, al menos en parte, por el proceso de senescencia celular. La senescencia celular, como lo vieron en la charla anterior, es una forma estable y permanente de salida del ciclo celular, la cual puede ocurrir en una variedad de respuestas, puede ocurrir a través de la senescencia replicativa, que no es más que el acortamiento de los telómeros, pero también puede ser encatillada como una senescencia prematura a través de la activación de oncogenes, aumento del estrés oxidativo y senescencia inducida por terapia, que es de interés para la presente charla. En términos generales, las células senescentes cumplen roles que son totalmente paradójicos. Por ejemplo, en un individuo sano pueden ser beneficiosos actuando como supresores tumorales, favoreciendo la cicatrización de heridas y participando en el desarrollo embrionario. Cuando estas células se acumulan, favorecen la promoción tumoral, el envejecimiento y el desarrollo de enfermedades cardiovasculares. Hasta ahora se sabe mucho lo que puede hacer la senescencia inducida por terapia en células cancerígenas, pero se conoce muy poco qué hace la quimioterapia, específicamente la doxorubicina, en células no cancerígenas como las células endoteliales. Ahora bien, las células senescentes son células que tienen características únicas, las cuales van a permitir su identificación. Acá nosotros observamos una célula normal, acá observamos una célula senescente, y como ustedes pueden observar, son células que son más grandes, tienen un aparato de Golgi prominente y tienen un citoplasma multivacuolado. Adicionalmente, estas células presentan características bioquímicas que permiten su identificación, como por ejemplo un incremento de la beta-galactosidasa asociada a senescencia cuando se encuentran en pH ácido, resistencia a apoptosis e incremento de algunos inhibidores dependientes de ciclina. Además de las características antes mencionadas, estas células tienen la capacidad de producir y secretar un gran número de sustancias que se conoce como fenotipo secretor asociado a senescencia, y es este el que puede explicar de mejor forma la relación que existe entre senescencia endotelial y trombosis. Estas moléculas bioactivas que se encuentran en el secretoma de células senescentes las podemos clasificar en 3 grupos, como factores de señalización solubles, componentes insolubles y más recientemente la presencia de vesículas extracelulares. Estas moléculas pueden actuar de forma directa o indirecta en el proceso de hemostasia primaria. Por ejemplo, en 2019 se reportó que células senescentes inducidas por quimioterapia a través del fenotipo secretor asociado a senescencia incrementaban la producción de plaquetas en el megacariocito y esto favorecía a la formación de trombo. Adicionalmente, también se ha reportado que células senescentes a través del fenotipo secretor asociado a senescencia podían incrementar la activación plaquetaria y esto finalmente favorecía la formación de trombo. Nosotros demostramos que células senescentes inducidas por quimioterapia a través del fenotipo secretor asociado a senescencia podían causar un incremento de la agregación plaquetaria. Sin embargo, se sabe poco de qué pasa en células que no son cancerígenas, es decir, en células, por ejemplo, células endoteliales senescentes, cuál es el efecto que pueden tener estas células en la función plaquetaria. Para ello, lo primero fue caracterizar un modelo de senescencia inducido por drogas en células endoteliales. En este caso, acá nosotros observamos la proliferación de células y acá observamos la beta-galactosidasa asociada a senescencia y se probaron concentraciones crecientes de la droga. En el caso de la proliferación, hubo una disminución relacionada con la dosis de la proliferación de estas células y en el caso de la beta-galactosidasa fue dosis dependiente su detección, lo que nos hizo poder escoger la dosis mínima necesaria para inducir senescencia, que en este caso es la que observamos en rojo. Posteriormente, la siguiente pregunta a responder era cuánto tiempo era necesario la incubación de la doxorubicina con las células endoteliales senescentes y para esto se probaron diferentes tiempos, observando que un tiempo de 72 horas era suficiente para detectar a las células senescentes, por lo tanto se estableció ese tiempo para el modelo que les estoy mostrando actualmente. Adicionalmente, también nosotros evaluamos la expresión de P21, en donde, como pueden observar, nuestro grupo tratado con la droga tiene un incremento y también medimos los niveles de laminin B1. Adicionalmente, nosotros teníamos que demostrar que nuestras células no estaban sufriendo un proceso de apoptosis y, como pueden observar, tanto nuestro grupo control como nuestro grupo tratado con la droga no está sufriendo el proceso de apoptosis. Finalmente, otra pregunta sumamente interesante que nosotros queríamos responder es qué pasaba con las células endoteliales y marcadores endoteliales. Para este punto escogimos un panel de varios marcadores endoteliales que nos permitían saber su estado, si estaban disfuncionales o no. Y como pueden observar, hubo disminución tanto de ENOS como de VEGF en nuestro grupo senescente y hubo un incremento en los niveles de nitrotirosina. Nuestras células senescentes se encuentran disfuncionales. Adicionalmente, analizamos el secretoma y observamos un incremento en la concentración de proteína y también observamos un perfil proinflamatorio. Todo esto nos llevó a tener nuestro modelo caracterizado, pero la pregunta que nosotros queríamos responder era cuál era el efecto de este secretoma en la función plaquetaria. Y para esto evaluamos diferentes parámetros de la función plaquetaria, en donde acá en este panel ustedes pueden observar el proceso de adhesión y en presencia del fenotipo secretor asociado a senescencia existe un incremento de la adhesión plaquetaria. Posteriormente, nosotros evaluamos el proceso de agregación plaquetaria. Acá en rojo pueden ver a nuestras plaquetas tratadas con el fenotipo secretor asociado a senescencia. Y cuando existe un estímulo subagregante de ADP, existe un incremento de la adhesión plaquetaria. En este punto, nosotros sabíamos que había un incremento de la adhesión y que había un incremento de la agregación plaquetaria. Para confirmar estos resultados, fueron evaluados dos parámetros para evaluar la activación plaquetaria y estos son la glicoproteína 2B3A y la P-selectina. Como pueden observar, en ausencia de estímulos subagregantes, no hay aumento de la activación plaquetaria. Sin embargo, cuando nosotros agregamos a nuestro fenotipo secretor asociado a senescencia junto a una concentración subagregante de ADP, logramos observar un aumento de la activación plaquetaria. Estos resultados nos llevaron a una publicación en donde nosotros postulamos el efecto que podía tener el fenotipo secretor asociado a senescencia en la activación plaquetaria. Posteriormente, nosotros nos preguntamos cuál era el efecto de las vesículas extracelulares en esta activación plaquetaria. Nosotros nos preguntamos si eran las vesículas extracelulares las que estaban causando un incremento en la activación plaquetaria. Y para esto el primer desafío era realizar la purificación y caracterización de las vesículas extracelulares. Acá nosotros observamos los resultados del nanotracking analysis, en donde, como ustedes pueden observar, tanto mi grupo no senescente como mi grupo senescente no presentan diferencia en cuanto a la concentración. Sin embargo, cuando se analizan las proporciones, nosotros observamos que las vesículas senescentes tienen un incremento en aquellas partículas que son menores a 150, a diferencia de su grupo control. Adicionalmente, hicimos los ensayos de microscopía electrónica y evaluamos a través de Western blot la presencia de vesículas extracelulares para confirmar que efectivamente teníamos vesículas extracelulares. Y hasta este punto teníamos una caracterización, sabíamos que nuestras células producían vesículas extracelulares y que éramos capaces de caracterizarlas. Ahora bien, la pregunta que nosotros teníamos era cuál era el efecto de estas vesículas extracelulares en las plaquetas. Para esto, lo primero que hicimos fue evaluar que nuestras vesículas no fueran tóxicas, debido a que era un método de purificación que para nosotros era nuevo, y evaluamos lo que fueron dos marcadores, calceín AM y la liberación de LDH. Como en ambos resultados se coincidió que las vesículas no eran tóxicas. Para medir el efecto de las vesículas en la activación plaquetaria, nosotros utilizamos los dos marcadores que habíamos utilizado anteriormente y nosotros observamos que en ausencia de concentraciones subagregantes o subestimulantes de ADP, nosotros encontrábamos un incremento en la activación plaquetaria. Estos resultados de P-selectina coinciden con lo que observamos en los resultados de la glicoproteína 2B3A, en donde solo la incubación de vesículas con plaquetas es capaz de aumentar la activación plaquetaria, y todo esto en ausencia de ADP. Gracias a estos resultados, nosotros postulamos que un paciente que recibe terapia anticancerígena desarrolla senescencia inducida por terapia tanto en células cancerígenas como en células no cancerígenas. Estas células no cancerígenas presentan cambios en su función, lo cual favorece a un estado protrombótico, y el fenotipo secretor asociado a senescencia puede activar a las plaquetas y estas a su vez favorecer la formación de trombo. Estos resultados fueron publicados ahora a principios de enero de 2026 y las proyecciones que nosotros tenemos en el presente trabajo es que por una parte se están realizando ensayos proteómicos, se están evaluando también ensayos de coagulación, y es de mucho interés saber qué pasa en células no senescentes, como por ejemplo a través de ensayos de migración y angiogénesis, y qué pasa con la permeabilidad endotelial en presencia de estas vesículas extracelulares o del fenotipo secretor asociado a senescencia. Adicionalmente, nos encontramos trabajando en un modelo matemático el cual busca estimar el riesgo trombótico con doxorubicina a través de diferentes fórmulas en las cuales aún estamos trabajando. Agradecimientos muchos. Este trabajo se llevó a cabo gracias a la colaboración de muchísimas personas que hicieron esto posible. Principalmente agradecerle a los que en aquel momento eran mis tutores, el profesor Rodrigo Muri, el profesor Nelson Brown y al doctorado en Ciencias Biomédicas. Cabe destacar que actualmente me encuentro como líder de un proyecto Fondesit de Iniciación 2025 específicamente, en donde en algún momento nosotros caracterizamos la sobreexpresión de un tipo de canales en células de cáncer de pulmón. Y nosotros postulamos en esta propuesta que cuando yo bloqueo mi canal con una droga, por ejemplo con ivabradina, voy a disminuir las propiedades tumorogénicas de la célula, y esto puede ser interesante para tratarlo como una estrategia novedosa en cáncer. Así que bueno, muchas gracias y quedo atenta a sus preguntas.
Muchas gracias, doctora Venturini, por su impecable presentación. Tenemos tiempo para preguntas. ¿Preguntas?
Buenas tardes, doctora. Excelente presentación, primero. Y tengo la consulta. Usted nos explicaba que cuando se hacía el experimento con en este caso el microambiente de la senescencia celular, sí, ese secretoma, hacía una activación de la plaqueta, ¿cierto? De casualidad, postularon realizar los fenotipos plaquetarios. ¿A qué punto llevaba la plaqueta este microambiente senescente? Por ejemplo, no sé si llegaba a una proagregatoria, una proadhesiva o una procoagulante.
Mira, sumamente interesante tu pregunta. En algún momento, cuando estábamos comenzando con todo el proceso de caracterización, evaluamos ver qué pasaba en la otra cara de la moneda, que era en el caso del secretoma de la plaqueta, pero en función a tiempos que teníamos que cumplir, nos centramos únicamente en lo que hacía el secretoma de la célula senescente en la plaqueta, y dejamos para futuros proyectos el tema de saber qué pasa específicamente con la plaqueta y su secretoma. Pero sumamente interesante, lo pensamos en algún momento, pero no lo hemos realizado.
¿Alguna otra pregunta? ¿No? Le agradecemos entonces a la profesora Venturini por su presentación. And now it is time to present our next speaker. The next speaker is Marcela Morales. Marcela Morales is a PhD candidate from the Faculty of Chemistry and Pharmacy at the Pontifical Catholic University of Chile, and she is performing her thesis under the supervision of Professor Camilo López. Please welcome Professor Mrs. Morales. The floor is yours, and I will turn on the computer.
Good afternoon. Thank you very much for being here. I'm going to present a part of my thesis, which is directed by my tutor, who is here, Camilo López. I'm a PhD student in chemistry in la Pontificia Universidad Católica de Chile in Santiago. And today I'm going to show you something that I'm still doing, but this is the final part of my thesis. I'm in the 4th year of the PhD. I'm about to finish, hopefully. So the name of this presentation is ditryptophan as a potential biomarker of protein oxidation in Cellular. And this is also under the supervision of Maria Fernanda Ormond, who is my co-tutor. So first of all, I want to show you what we do in our lab group. So we work with proteins, and we study the oxidation of these proteins because, as you might know, proteins are the most abundant molecules that we have in our bodies, and they are exposed because of probability and also susceptibility to reactive species. Depending on the reactive species and also the protein and other conditions, we can have several consequences, as you can see here, for example, unfolding or side chain modifications. But today I'm going to talk about cross-linkage because the two biomarkers that I'm studying are products of the dimerization. So here I'm showing two of the two dimers that I'm studying. One is the tyrosine, which comes from the oxidation of tyrosine, and the tryptophan, which comes from the oxidation of tryptophan. So why they are important? For example, in the case of the tyrosine, we can see that there are several research that have shown that the tyrosine has been found in tissues from Parkinson's disease, forming part of the dimers that are making the aggregates. And in the case of the tryptophan, I want to point out this because this is the novelty that we are proposing as a biomarker. So here we can see an early study that was kind of like the first study that showed the presence of the tryptophan in proteins. So here we have SOD dimerization because of its peroxidase activity. So they proved through LCMS analysis that the tryptophan was making the dimer of this enzyme and as a consequence of its oxidation. So another study that I want to show you is the oxidation of A-lens proteins, crystallines. They showed, as you can see here in the picture, that after they hydrolyzed the crystallines, they found the tryptophan dimers as part of the dimers that were forming the aggregates that were part of these disease cataracts. So the tryptophan dimer was part of the covalent aggregation of this disease. So now that we know the biological relevance, with my thesis we are trying to cover two main things that are kind of like a weakness in this type of study. So here we have a low availability of standards and also limited identification in vivo. So first I'm going to start talking about the methodology that we use to produce this dimer. So this is an enzymatic way that we use. It's a very known enzyme, which is horseradish. It's a vegetable peroxidase that comes from plants. So here we have the mechanism. I'm not going to explain this part, but I'm going to show you that because of this mechanism, we can have two radicals of the reductant. So because of this type of mechanism, this enzyme is used to produce different dimers, trimers, and also polymeric structures. So first of all, what we can know from this enzyme is the most used method to produce the tyrosine. So we thought that if this is used to oxidize tyrosine in order to produce the radicals and then the dimer, so why not using the same system to produce the tryptophan? So how we proceed when we started this study? Well, we studied the pH for the enzyme. Also, we measured tryptophan and hydrogen peroxide consumption. We did a chromatography separation because the dimers, we wanted to concentrate the dimers to be able to observe them in the LCMS identification, and we also did a docking analysis. But today, I want to show you just the LCMS results because that's the beginning of this study, and then we're going to talk about Cellular. So here are the two molecules that we are identifying through LCMS because of the delocalization of the electron in the tryptophan radical. You can have several isomers, so we are following these two fragmentations. And here we were very happy because we found the dimers in the enzyme, as you can see here, the signals that were not different regarding the pH. So we observed several isomers for the two transitions that we were following. And also the highest intensity was in basic conditions. So now here we have the first thing that was the pattern of dimers that were obtained from this system. And we were happy to publish these results last October in case you want to know more about these results. So now that we have the dimer pattern, and also that I showed some in vitro identification. In order to be able to call the tryptophan as a biomarker, we needed an in vivo identification. So this is the moment for C. elegans. So C. elegans is used as a model in oxidative stress and aging studies because of several things. I'm going to show just 3 of them. 60% of the genes are homologous to humans. Also, like a short life cycle allows in research to do aging studies because you can observe the entire life and also make some predictions. And it's easy, not really easy, but it's a low cost in terms of infrastructure and maintenance. So we started using C. elegans in this way. This is something that we kind of like made up at the lab because we needed a certain amount of protein to be able to identify the dimers. Because C. elegans is usually used with less amount of protein. So what I did, I cultured C. elegans into wild type in 2 stages of life. So 55 hours and 20 days. I did it twice to have 2 independent replicates. Here you can see how the pellet looks like. And after that, I used the sonicator to release the protein and to break all these cell extractors. And I collected the supernatant to measure the protein through BCA protein assay to be able to be sure that I was going to hydrolyze this amount of protein to normalize the results that I was going to have. And then I identified the dimers through LCMS. So these are called, for me, the first results because then you're gonna see what happened next. So here we were also very surprised when we observed that we actually got some signals for the tryptophan. So as you can see here, we have signals for 203, the first fragmentation, and we can observe 2 of them like very resolved and also with a higher intensity compared to these 2 small ones here. And also the intensity was higher in the age ones. And for the second transition, we also observed signals, but they were not as resolved as 203 fragmentation. So how did we know that these were the tryptophan? Because it's a complex model, so you can have this response for this transition from something else. So that's why we had first the pattern, right, that we got from the enzyme. So here you can see that we have like a comparison between the standard that we prepare with the enzyme in order to have the retention time and also the range of the dimers. So this first part was important to be able to identify, to be sure that we were identifying the tryptophan. So we wanted to know if the tryptophan was really formed in protein oxidation during natural growth. Also, if it's increasing as a consequence of age, and what about another cross-linking product? So here at the second part of the study, so I did this in Brazil where I was in my doctoral stay. I did the same thing, but I had to do some changes because each lab works with its own, procedure. So here I just want to point out that I had to hydrolyze less protein because I needed to normalize according to all the pellets that I got. And I also added 2 stages of life more. So after this, I also measured basic parameters of C. elegans and the fluorescence of 2 reporters, GST4 and SOD3. Okay, so I'm going to show you the second result that I got. So first, I'll show you the size of C. elegans, that as you can see here, they reached a certain size. They're not going to grow as they age, so they just reached a certain length of the body, like in 72 hours. So it was accurate, the result that we got, because it didn't increase the size when they were aging. So here, because I know that everyone wants to see the worms, so we have here the young ones. I don't know if you can see that they are, compared to the old ones, they are shorter and also thicker. They are thicker, the old ones. So here we could see that there are no changes in the length of the worms after 10 days of life. Then I started measuring bends, which is the way that we measure movement in C. elegans. So here you can see the young ones, they move like all the time. So what we are measuring is just how they do this. And the old ones, you can see that they barely move. They're very slow because they old. So here we have that the worms get significantly slower as they age. So that's also in agreement with the research which have been published. So the result for GST4, which is an enzymatic enzyme that C. elegans uses as a part of the detoxification process. So we can see that it's increasing as C. elegans age. So we see here, for example, one of the images that I analyzed. We can see in 55 hours how the fluorescence looks like, and then when they age, you can see that it's increasing, right? So here we were able to say that GST4 expression increased with the age of worms. So the second enzyme that I was following was SOD3. So here we have the first stage, which is the young ones. So I was expecting to have the same behavior as GST4, but it was a surprise, at least for me, because this was the first time that I started to work with this type of strains. So here we see that the fluorescence, even if it's spread in comparison to what we observe here, I was measuring just the head to be able to have comparison with the fluorescence in early stages of life and after the age. So here we observe that this fluorescence was lower. And when I was trying to explain this behavior to be sure that it was something like in agreement with the behavior that has been reported for C. elegans, I found that because C. elegans have several SOD, 5 of them actually, this one especially is triggered by oxidative stress. So it's kind of normal that we have less fluorescence as C. elegans ages because they use the other SOD enzymes to like aging mechanism defense. And the tryptophan results, so here is important to point out that we have like a different behavior. As you can see here, for 55 hours, like we barely observe signals. If you remember what I showed at the beginning, that was different. And also for 10 days, yeah, we started to see some of them, right? And for 15 days, was not as clear as we thought it was going to be. So that was not cool for us, right? Because we thought that with the first result that was going to be like a conclusion. But in this case, we can say something important, that even if we didn't observe the same behavior at the beginning, we still are seeing signals, right? Just maybe what happened is because I had to hydrolyze less protein, or maybe a dilution factor that affected the results, but we still are observing this signal for the tryptophan. So because of this, we also wanted to check some other products, especially because in our group lab, they have been working with several oxidation systems with several proteins. And here I wanted to show that, for example, in this publication from our group lab, lysosine was exposed to peroxide radicals, and they also found heterodimers, tyrosine tryptophan. So because of this, I also looked for this dimer in C. elegans, and we found nice results for all of these stages that I analyzed. As you can see here, we observed like nicer signals for YW or tyrosine tryptophan dimer. So we can say from these results that we observe these dimers in all the stages of life that we analyzed. So I was happier this time. And summarizing, we got the solution to measure C. elegans from the enzymatic system. So we had the pattern of dimers, since the tryptophan has no commercial standard, so that was good for us to be able to compare what we got in C. elegans. And the formation of the tryptophan was observed in C. elegans as part of their natural growth. SOD3 expression was lower during aging, unlike the GST4 expression, which increased. And signals of heterodimers, tyrosine tryptophan, were also observed. This is not like a concluded study, but those are the results that I have obtained so far. So what is ongoing or the perspectives that I have to finish soon is N2 wild type was analyzed in all these stages of life, but because I was in Brazil with a group that works with C. elegans, I added 5 more strains that are connected to aging and also oxidative stress, and I was able to collect 2 days and 10 days for all of them. And finally, I exposed the N2 wild type to several oxidants like paraquat. Finally, I want to thank my scholarship and also the project that is connected to this topic, the opportunity for an oral presentation to the summer school, and also my group lab, which is full of nice people. My tutor, that is the person that is driving all this group. There are people that have gone already, but they're very important in the research part that we as PhD students do. Thank you very much.
Thank you, Marcela, for the excellent presentation. We have time for some questions. Who?
Yes, very interesting presentation. How did you perform the hydrolysis of C. elegans protein? Was it acidic hydrolysis or?
Yeah, acidic hydrolysis with methylsulfonic acid.
Okay, but tryptophan usually is not stable with acid hydrolysis, so how did you stabilize it?
Yeah, we have a way in our lab. We use tryptamine. So we use like a high amount of tryptamine to be able to hide tryptophan from the oxidation or the degradation that can happen in acidic conditions.
Okay, I didn't know. Thank you.
Anyone else? Is a question? No? Okay, thank you, Marcela. And now, according to the program, we have coffee break. So yes, we can enjoy coffee break, and which is sponsored by Prime Lab. So please enjoy.
termina, tiene que llevar a Enero.
Bueno.
Es la otra.
Sí. Sí, por supuesto. Voy a citar son 40 minutos. Sí, como en 40, 45 minutos. Ah, sí. Claro. Súper. Gracias. Everything okay? Ah, yes. Perdón.
Basic hydrolysis with an AOH. Normal acid hydrolysis, you get all aminoacids except the profile. But in this case, they are doing something special to delicate the profile.
Yes. Yes. They are. Yes. Yes. This is a collaboration with the group of candidates in Denmark.
Yes, I saw that.
Yes.
Well, I will ask you.
I was once in his lab.
Yes.
They get the YouTube of my
Yes.
And also they quantify
What they should do to make sure is the accurate mass. You know the exact mass with 3 digits or 4 digits after 0. You know exactly the structure.
Yeah.
Because with the just refinements, as she said, we can do something else. And then looking at just the revolution time, or it's an indication, it's a good indication already, but the accurate mass, or then she will need to perform the analysis with an arbitral or something else. The accurate mass will be the formula.
Yes.
The formula. That would be useful.
Yes.
And I guess the guy in Denmark doing that is also proving by the accurate mass or Denmark would be useful also that you have to purify.
Yes. Maybe in a case of one day, it should not be so easy to modify.
That's
To get the minimum.
Or with the silicon la teoría, pero
No, él estaba abajo.
Okay, welcome back. Okay, it is a pleasure for me to introduce Professor Alejandro Corbalán. Professor Corbalán is a full professor of medicine and principal investigator at the Advanced Center of Chronic Diseases at the Pontificia Universidad Católica de Chile, and an international recognized researcher in oncology and cancer biology, particularly gastric cancer. He has completed advanced training in the United States and Japan and led numerous national and international research projects. He has authored over 130 scientific publications. His work has been recognized with major awards, including Chilean Oncologist of the Year and the Agony Award for Innovation in Health. Professor Corbalán, welcome.
Good afternoon, it's a pleasure to be here. Thanks for the organizers for this invitation. I will talk about this H. pylori-related senescence. The title of my talk is The Dual Role of Long Non-Coding RNA, KCN1, OT1, and H. pylori-induced senescence in the development of gastric cancer. This long and complicated title will be split in several parts, so for you to understand. So first of all, gastric cancer is a major problem. This cartoon shows that gastric cancer in males is among the top 5 tumors in the world in terms of incidence as well as mortality, with representing 10%, 9.1, close to 10%, to the mortality in terms of cancer-related cases in the world. In females, it's less frequent. In terms of incidence, but in mortality is again top 5 in the world, with 6% of the cases in the world in 2021. The distribution of gastric cancer in the world is mostly in Asia and Eastern Europe and Latin America. And in terms of around the Pacific Rim. So that's why I like to put this map in different orientation, highlighting that in South America, it's mostly the Pacific coast. It's not a problem in the Atlantic side, rather in the Pacific side. And Guatemala, Colombia, Ecuador, and Chile are the most important places in Latin America. And in the other side is Asia, as I mentioned, so it's a disease in terms of geographic location around the Pacific Rim. What's the relationship between these two areas? I don't know, but in some way it's this is such a common topic across the ocean. The cancer as a problem is an emerging problem. Even if Chile is almost, it's number 2, but close to cardiovascular diseases. But I said emerging problem because it's expected to increase in the next years. So I'm showing here this is a publication that was published in Revista Médica Chile 2023 by a group of Newly region, and show the expectation of the increase between 2022 and 2045 in the world and also in Chile. As you can see here, it will increase from 20. I don't see very well, but significantly in between 2022 and 2045. In terms of incidence and mortality. And as you read here, gastric cancer is among top 5 in both scenarios. So gastric cancer is not an important problem, it will become an important problem in upcoming years. What is the pathogenesis, the etiology of gastric cancer, and how it's classified in terms of molecular markers? This cartoon shows a molecular classification in which there are 4 types. The EBV related, which EBV is the Epstein-Barr virus, which is endemic infections, but is related to around 8.8, almost 10% of gastric cancer. In Chile is quite high, more than 15% of cases. Second subtype is the genomic stable, which is almost 20%, which is quite paradoxical because this is most resistant to chemotherapy and but does not have any mutations or very low rate of mutations. So it's something is going on here. The other one is microsatellite instability, which represents almost 22%. And this is mostly related to failure of repair mechanism in tumor cells. And the fourth one is chromosomal instability, which represents almost 50%, which fits very well with the rate of p53 mutation, which is about 50% of the cases, gastric cancer cases, and also related to H. pylori. So there's a good correlation with etiological agents related with gastric cancer. As I mentioned, the etiology of gastric cancer is related to environmental factors on one side or genetic factors, and these are two different etiologies that create two different pathways. As I'm showing here, the genetic pathway is mostly related with heritage and does not have any precursor lesions. Usually it tends to occur in females more than males and at young age, around 35 years. And mostly related with run within families. And what is important, that's also related with hereditary breast cancer. So this even is a syndrome, breast cancer tumors. But in the other side, and the most common type is environmental, associated with is mostly related to H. pylori, and also in some risk factors like diet or smoking, also wine or alcohol consumption, but it's associated with this long stepwise cascade of different lesions that's called gastric premalignancy. And this is important because this is an opportunity to early interventions and to understand what's going on between normal gastric tissue to tumor gastric tissue. A difference, the opposite is the hereditary associated with does not have any precancer lesions. So in this case, we have this stepwise cascade that's atrophy, intestinal metaplasia, dysplasia, gastric cancer, and this is called intestinal type gastric cancer, or mostly in molecular terms, the chromosomal instability, p53 mutated tumor type, and this is called gastric malignancy. This idea of the premalignancy with these stepwise lesions was developed by Correa, who passed away at the end of last year, a couple of months ago, at the age of 98. He was a pathologist and also epidemiologist who, rather to discover these lesions of atrophy and intestinal metaplasia, organized these lesions that were well known at that time, but nobody thought there was like a stepwise progression. So he's the one who proposed this atrophy, which is multifocal, I'm showing here. It's only one lesion, but multiple lesions that evolved to intestinal metaplasia and then to dysplasia. And several years after that, which was described in 1975, H. pylori was discovered, and subtypes of H. pylori were related with the development of the cascade, and others was unrelated to the development of the cascade. And this cartoon shows the difference between this non-atrophic and the multifocal atrophic, clearly showing that one is multifocal lesion and the other one is localized. This cascade is not linear, it's not simple, it's back and forth progression. And this is a publication from a group from my institution, Catholic University, that is the follow-up for patients with atrophy and intestinal metaplasia, and with a follow-up for more than 6 to 8 years. And these lines try to show the dynamics of this natural history or natural evolution of these precancer lesions. As you can see here, this all serum is low grade of atrophy, and most of the people, most of the patients stay as low-grade atrophy or acero. This is a classification that is called operatively or assessment. It's a way to understand between clinical studies. But if you move up, lesions with more advanced atrophic arthritis or even intestinal metaplasia tend to progress to higher degrees of intestinal metaplasia or even to high-grade dysplasia or gastric cancer. So this cartoon is trying to show that there is a slight progression from low-grade lesions to high-grade lesions, but could be in the other way too. That's why the opportunity of to eradicate H. pylori, because at early stages this lesion can be reversed and stop the progression. But at high levels of gastric malignancy, eradication by H. pylori is not useful. On the other side, senescence has been studied in the gastrointestinal tract, in the upper gastrointestinal tract, since 2002, more than 20 years ago. This was the first publication, and this shows something what is quite interesting, that in normal and in gastric there is low grade of senescence based on beta-galactosidase marker. But if you see in the lesions that correspond to gastric malignancy, what I've shown here, different location of the stomach, there is a high degree of senescence which tends to disappear in the progression of progression to gastric cancer. So this was published in 2002. This is nothing related with H. pylori. Just senescence staining in the human gastric mucosa. And in 2021, almost 20 years later, a group of researchers identified that the infection with H. pylori was were able to induce senescence in the gastric mucosa, in the human gastric mucosa. And as you can see here, two slides are showing the presence of this staining. In this case, Sudan black B, which is a marker that can be done in paraffin embedded tissue, which are cleaner samples. And here is the level of this Sudan black B staining in lesions that are H. pylori negative, which means normal gastric mucosa, or H. pylori positive, which is in some way one of the some steps in this gastric malignancy cascade. If we go into details of this progression, rather than of the different steps of the cascade, rather than H. pylori, you see that in chronic gastritis, non-atrophy, there's low grade of senescence, but in atrophy, there's increased grades of senescence, and then it came down in intestinal metaplasia and then in dysplasia. So that's some people call the loss of senescence in the progression of the gastric lesions, or paradoxical role of cellular senescence in cancer development, or more simple escape from senescence. So the question here is, it seems that in premalignancy, senescence is part of the phenomena of the premalignancy, but in terms of, which means cell cycle arrest. But in gastric cancer, there's a loss of senescence or escape of senescence, because gastric cancer is a hyperproliferative lesion. So in these two papers, what I'm showing is that senescence is part of the premalignancy, but it's lost in cancer stages. So a couple of years ago, we award a grant, a fundraising grant, to study the role of senescence in the premalignancy, and we focus on genes, tumor suppressor genes that were related to the cell cycle, mainly RB1. Initially, we considered P16, P21, and phosphatase, acid phosphatase as a regulator of RB gene. Our approach is not related to coding genes rather than non-coding genomics. And the reason of that is because after many efforts and many years of effort looking for mutations in tumor samples, this image what shows that this scarce number of mutations of a panel of them. So this low mutation rate in the genes that are related with cancer. So that's in some way means that there's something else that it may go on that damage those genes, but not directly in the coding sequence. So our focus is that probably we're looking the tip of the iceberg, looking for the coding genes, the genes that produce proteins by RNA, messenger's RNA, where something below, maybe below the water, or below of the coding genes that is regulated or dysregulated these coding genes. And this is what's called non-coding genomics, which has been developed by all the sequence and bioinformatic analysis of genes that, as I'm showing here, are sequences that are 80% transcribed. 80% of the non-coding genomic is transcribed into RNAs, but not codified into proteins, not transcribed into proteins. And 40% of genetic diseases are associated with polymorphisms located in non-coding genomes. So there are some issues that show or suggest that non-coding genomes in some way regulate coding genomes. This is a classification that's quite simple between what is a small non-coding RNA and long non-coding RNA, which is 200 nucleotides. The size less than 200 is called small or short RNAs, and more than 200 up to almost 100,000 kilobytes is considered long non-coding RNAs. And there are several subtypes in both families. And probably the most important one in the case of small non-coding RNA are microRNAs. So it's my focus of my talk. So how non-coding genomics or non-coding RNA regulate coding RNA? Basically, there are two ways. One is shown here that microRNAs binds to these three untranslated region of the messenger RNA and induce endonucleotide degradation, or non-coding RNAs binds to the 5 untranslated promoter region, inducing DNA methylation. So these are two ways. This is DNA methylation. This is post-occasional down-regulation of filtration. So this is one of the ways how they interact. But there's another way in which microRNAs bind these 3 untranslated regions. But at the same time, they can be bound by non-coding RNA by a sponge. So it means that a microRNA has two possibilities to target messenger RNA and degrade the transcript, or be sponged by non-coding RNA and be inactivated. So it's like a tuning of what happened with a messenger in terms of transcription. And this competence between messenger RNA and long non-coding RNA for the same microRNA is called the competent endogenous network. And they call the Rosetta Stone because Rosetta Stone is a stone that has a speech written in 3 languages, ancient Greek, and 2 types of ancient Egyptian, and because this was a well-known language, opened the understanding of the 2 scripts that were unknown until the time. And stone, Rosetta is the name of the city where this stone was found. So that means there's 3 languages that they talk each other, and that's the concept of competent endogenous network. As I'm showing here in this slide, is, for example, one biological phenomenon, which is epithelial mesenchymal transition that is regulated by several genes, which are in black here. in black circles. These are regulated by microRNAs, and microRNAs are regulated by long non-coding RNAs. So this is a huge network, which I'm showing here again. This is a epithelial mesenchymal transition, coding genes, microRNAs, long non-coding RNAs. So that's why this is a network, how cells regulate gene function. Outside the gene by itself, the coding gene by itself. And may explain the low rate of mutation in tumor cases because probably the damage is in microRNAs or long non-coding RNAs. How this competing endogenous network can be studied? One of the approaches is to study the co-expression between a long non-coding RNA, I'm showing here, with a coding RNA, which is brand X1 as an example here, and they have a not perfect but quite high correlation between expression, because the concept is that I have a long non-coding RNA responding microRNA, messenger can be expressed and they are not degraded. And as you can see here, more long non-coding RNA expression means more messenger RNA expression. So this is one approach. The second approach is to use these dices after small interference, in which you use a small RNA that inactivate expression of long coding RNA, which I'm showing here. This is negative control. This is with a cocktail of 3 probes that inactivate the long non-coding RNA, and the consequence is that this messenger RNA here is also downregulated. It means that there's some microRNA in between that is has been sponged by a long coding RNA and now is free and can inactivate the messenger RNA. So this is the second way to study. And the third one is to use bioinformatic tools to predict bindings between long non-coding RNA and microRNA, and then use luciferase binding assay to demonstrate that indeed there is a binding by luminescence that is lost when you use a probe that's mutated, showing that this binding does not exist. So here we have binding, here we don't have the binding. And in this case you use double binding or between long non-coding and microRNA and messenger and microRNA. So it has to be in both directions. So these are the 3 ways to study this competing endogenous network. So moving back to senescence, this initial collaboration that we have with Nelson Brown, who led us to use palbociclib with all the protocols behind to look senescence in gastric cancer cells, what I'm showing here, and these are the difference in terms of senescence. And then we also did with beta-galactosidase in gastric cancer cell lines. And also we were able to stain tumor tissues, in this case paraffin-embedded tissue, and tumor and non-tumors. You can see here there's no senescence here, and here there's some island of senescence in non-tumor gastric mucosa, in some way confirming what I described before, that senescence exists in non-malignant tumor, part of the gastric malignancy, and is lost or is absent in tumor cells. With that, in collaboration with Andrew West Group, we infect the cells with H. pylori, and we evaluate senescence in different cell lines, AGS and NCI-N87. This is control, and this is H. pylori, and you can see here there's some staining showing that we are able to induce senescence by H. pylori infection. The long non-coding RNA that we choose was selected in a previous project using bioinformatic tools. This is the work of Ignacio Wickman, who combined a tool that's called New Work that select almost 400 non-coded RNAs based on microRNA 335 that were the one that were started at that time. And also we used the Cancer Genome Atlas, which is a huge repository of transcription data, methylation data, protein acid data of 33 tumor samples. In this case, I'm showing gastric cancer, in which are at least 450 available tumors for analysis. So it's a huge kind of tumor bank, digital tumor bank, to explore hypotheses. So in this case, we identify 19 non-coded 91 upregulated non-coded RNAs. For this cross-match, we identified 4 microRNAs, and we choose one, which is this one, EKCNQ1.1, for one reason. This is the longest long non-coded RNA in human genome. This is the longest one. It's almost 92,000 nucleotides. And it's a monexonic gene, which has at least the capacity to bind 700 microRNAs, which means 30% of the microRNAs can be bound by this long non-coded RNA. So it's a huge gene, but in terms of competing endogenous network, it's a huge sponge that can bind almost 1,000 of microRNAs. So that's why this is our candidate for our studies. So we did expression in H. pylori infected cell lines in this gastric cancer cell lines. As you can see here, there is an increase of expression of this long non-coded RNA and RB1, which is a gene that is a key gene in senescence. In this cell line, this was also tested in other cell lines. But also we found that for RB, this was not a consistent result. You can see here that at protein level, there's no difference between infected and non-infected cells. But there are significant difference in terms of phosphorylated RB, which is the active one, the one that is regulated by acid phosphatase. And as I'm showing here, there's a balance between senescence and proliferation that is mediated by RB1, but depends the activity of acid phosphatase, PP1A and PP2A. So basically this balance, which is where RB is basically dependent of the phosphorylated state of RB1. So as I'm showing here, there's a clear difference between the H. pylori infected and non-H. pylori infected based on urease B, which is a marker of H. pylori, and the presence of phosphorylated version of RB. In this slide show the levels of acid phosphatase in terms of presence of H. pylori. You see it's upregulated. These are some immunofluorescence for H. pylori and acid phosphatase. This was also correlation with the group of Andres and by Christophe Collet. And this is the assay with the Dyson, in which we were able to downregulate KCNQ1 here. And this is the downregulation of phosphatase 1A, which means that has to be a microRNA because we are downregulated the long non-coded and the messenger is going down too. So we did this informatic analysis, and here we identify one microRNA. We identify around 5 or 6 microRNAs, but we choose one because we have to choose the best one because we have to do this double assay for the long non-coding side and for the messenger side. So we choose this one, which is in the process of being set up in the laboratory for the luminescence assay, which is microRNA 4319, and this was based on the novelty because this is a totally unknown microRNA that has not been described. So we are focusing on this microRNA in order to show that this is the microRNA that links genes related with senescence and genes that are induced, like induced by H. pylori. So this is an interesting idea because in some way we are starting in the environment with H. pylori, inducing long non-coding RNA, and through competing endogenous network we are playing with this balance between senescence and proliferation, which is probably something that we and others showed before that is lost in the premalignancy. It's acquired at the beginning and then it's lost. And to finalize, and that's the title of the project, and this talk is the dual role, is as a pilot data of the project, we found that the proportion of this long coding RNA was part of the cytoplasm and part was in the nucleus. So it's not 100%, but it's a tendency that there is some difference in the proportion of subcellular localization. And in the cytoplasm, it's related with competing endogenous network because they are messenger RNAs. But in the nucleus, it has a totally different role. And this is the first slide that I show you, that in the nucleus they induce the remethylation. So they can shut down genes related with cell cycle regulation. And this is the work of Francisco Berrocal that we analyze here. These samples of atrophic gastritis, intestinal metaplasia, and gastric cancer. And you can see here there's a clear difference in the, in this case, nuclear localization between atrophy, intestinal metaplasia, and tumor gastric cancer samples, showing that indeed there is a predominance of nuclear localization of this long non-coding RNA in the gastric cancer stage, in contrary to what happened in premalignant stage, which predominate the cytoplasmic localization. And these are the monofluorescence. These are the results of the monofluorescence, and also is also related with the H. pylori staining, which is not that significant, but this is probably because the role of H. pylori is important at the beginning of the gastric premalignancy and then it's lost. So this is our working model in which we have this atrophic gastritis here, H. pylori, that is induced expression of KC-Q191. The same happened in gastric cancer, so it's up-regulated all the time. But in the premalignancies in the cytoplasm, binding or sponging microRNAs that in some way led the upregulation of this gene that induce senescence. But in gastric cancer cases, go to the nucleus and induce remethylation. That's something that is going on now in the laboratory. Actually, PT acid phosphatase has a promoter that is methylated. So probably now we're in the process to perform these assays. So I think that at the early stages, gastric cancer, premalignancy, H. pylori induced KCN, promoted senescence using this competing endogenous RNA network. But later stages of premalignancy, maybe gastric cancer itself, KCN translocated to the cell nucleus and potentially downregulate senescence associated genes and lead the cells to enter into proliferation. So this is my group. And Gonzalo La Torre, who's the one who ran the clinical work, and international collaborators, and particularly Pelay Correa, whose foundation passed away 2 months ago, and Fondesit and Facultad, who's handling grants that support this work. Thank you.
Thank you, Professor Cortolán, for your excellent presentation. There is time for some questions. Who wants to start? Any question? Okay, so maybe we can continue the discussion after in the poster session. Thank you. Okay, it is now my pleasure to present our next speaker. Is Professor Andrew Quest. Professor Quest is full professor at the Faculty of Medicine and principal investigator at the Center for Studies in Exercise Metabolism and Cancer at the University of Chile. Professor Quest obtained his PhD in biochemistry from the Federal Institute of Technology at Zurich and completed postdoctoral training in the United States. He has authored more than 200 scientific publications, and we would like to thank Professor Quest for being with us. Something strange is going on. Okay. Please, Professor Quest.
That's the one. Okay. Well, thanks for the introduction, and also the possibility to present here today as the last speaker of the day. I figured probably you've had enough of talks about senescence, probably already becoming slightly senescent. So I'm going to talk about something different, particularly about this center here, SENSA, what it means and the transitions it's gone through and what we hope it might become in the not too far future. I thought I'd do so because it would be interesting, particularly for young investigators and students to know what's happening now. So, as I said, we are part of the center that at this point is called Center for Studies on Exercise, Metabolism and Cancer in the Faculty of Medicine at the University of Chile. So, we've been in Chile now 26 years, got here in 1999, and built there basically from scratch. The laboratory has become known as the Laboratory of Cellular Communication, rather successful. Many people have gone through the lab, and not all of them are listed up there, and have gone on to have independent positions, and so on and so forth. The lab is run essentially by two PIs, Lizette Lade and myself. Lizette Lade that works on astrocyte neuron interaction, so this part here, and I work on the cancer-related section here, and recently a young investigator joined our lab, Lorena Lobos, and she also addresses cancer-related questions. But the focus will be on this part here, the SENSA facilities that are run by Cecilia Zuniga and Hervé Camus. Okay, So the title is Center, Past, Present and Future. As you can see here, this is how it all started. Initially as the Center for Molecular Studies of the Cell, thus CENSE, and this was actually a FONDA project that was funded from 2002 to 2012 and studied obviously molecular mechanisms involved, for instance, in cell signaling, amongst other things, and it was a large group, roughly around 200 people at some stage. However, in 2012, funding ceased to be, and the center would have sort of disappeared.